Welcome to the channel Bharat's Pharmacy. So in this video, I'm going to explain about the IMViC test in brief and a quick outline about this IMViC test. So this is one of the biochemical tests used in the identification of bacteria. So there are various identification techniques like staining and biochemical test. A small part of the biochemical test and very widely used test for the identification and differentiation of Enterobacteriaceae is called IMViC test. What is this IMViC? How it is derived? IMViC, it stands for indole production test, and M it stands for methyl red test, and V it stands Voges-Proskauer, and C it stands citrate utilization test. So the term IMViC, it is an acronym. It is derived from the indole, methyl, and Voges-Proskauer test, and this test we are performing in citrate agar medium. So that is why that I letter it is in small letter it is mentioned, and to form the term IMViC. So to differentiate the indole and the small I, so the I it is made it in small letter. So that is why this test, the acronym it is called IMViC test. So all these tests we are performing to differentiation and identification of enteric organisms. So I didn't explain all the enteric organisms. For your simple understanding, I have taken three organisms like Escherichia coli, Klebsiella pneumoniae, and Enterobacter aerogenes. So these three organisms I'm going to identify and differentiation by using these biochemical tests. First, we'll look into the indole production test. So this is one of the indole. So it's one of the metabolic degradation production product of amino acid, tryptophan. So in this test, an indole production test, the basic principle is the main use of IMViC already we discussed. It is used for identification and differentiation of the enteric organisms. Like enteric means it is intestinal organisms, and it's very important for controlling as well as the controlling of intestinal infections because of the food contamination and water supplies. And this test is mainly used for differentiation and identification of enteric organisms. And bacteria that possess the enzyme tryptophanase are capable of hydrolyzing and deaminating the tryptophan with the production of indole, pyruvic acid, and ammonia. So to perform this particular test, we are using a medium called a SIM agar. This SIM agar, it is rich source of tryptophan. And again, this SIM, it is having another introduction and interesting explanation. This same medium can be used for three different purposes. So one, it is used for the sulfide protection test identification, and also the same media it is used for the indole production test. In this media, it is also used to confirm the motility. I mean, the presence of flagella can be confirmed by using this SIM agar medium. But in this, I am explaining only about the indole production test and its principle behind in this SIM agar medium. So SIM agar, along with the additional requirements, we need to have the coax reagent, otherwise it's called acrylic reagent, the composition of para-dimethyl aminobenzaldehyde, concentrated hydrochloric acid, as well as amyl alcohol. So in this procedure, we take a medium of SIM agar medium and then inoculate the organism, test organisms which you want to identify. So in this purpose, I am taking Escherichia coli for your understanding. And after the inoculation, okay, incubated 24 hours at 37 degrees Celsius. After 24 hours, when you add the 0.5 ml of coax reagent around the inner wall of the test tube and examine the upper layer of the liquid, and based on the color change, you can say whether it is indole positive or indole negative. So what exactly it is happening in during these 24 hours? So as I told you already, the SIM agar, it is containing a rich source of tryptophan, and in the presence of water in the medium, when you introduce the organism, only few enteric organism is having the ability, it is able to secrete an enzyme called tryptophanase. So this tryptophanase, it acts on the enzyme, this tryptophanase enzyme, it acts on the tryptophan and it produces indole and pyruvate and ammonia. So at the end of 24 hours, so when you add a coax reagent, the composition is nothing but para-dimethyl aminobenzaldehyde and amyl alcohol and hydrochloric acid. So it causes a dehydration reaction, and it forms a quinidal red-violet compound. And this compound, it produces a red color, cherry red color on the inner, the ring portion of your indole medium. So on addition of coax reagent, a cherry red color is observed. On the interface of the reagent, it indicates the presence of enzyme tryptophanase. When there is no color, it indicates that there is no tryptophanase production. And from this tryptophanase production, we can confirm what genera it is. And this is what the actual test tube it looks like. So the left side one, it's a positive for indole production test. By the time when you add coax reagent, the amyl alcohol and hydrochloric acid, it helps in the extraction of the indole from the whole medium. So most of the tests, if you see when you add an ingredient or the reagent, the whole media will get changed in the color. But this indole production test, the indole is extracted by amyl alcohol and hydrochloric acid, and it brings to the solvent layer, and that is the reason it produces cherry red color only on the top portion, the inner layer. And when it is not producing any color, that indicates that indole is not produced. That means the organism doesn't have the ability to produce an enzyme called tryptophanase, and we can say it is the indole negative. So E. coli is one best organism. It is example for indole positive organism. So we can identify, but still only one test, it won't be sufficient for confirmation. So we can go for the other test so that you can confirm whether it is belongs to Escherichia coli or not. So indole positive one organism is Escherichia coli, and indole negative both Klebsiella pneumoniae and Enterobacter aerogenes. It won't give any positive reaction in the indole tryptophan agar medium, because those two organisms doesn't have the ability to secrete an enzyme tryptophanase. Now we'll come into the second test, an IMViC methyl red test. So in this test also, we are using for differentiation and identification of the enteric organism. In this case, to determine the ability of organism to utilize the glucose, and it produces a stable acidic end product. And only few genera of Enterobacteriaceae is able to produce a stable acid after the utilization of glucose. And that is the major concept, a major principle behind this methyl red test. And some bacteria, it utilizes the glucose and it produces the stable acids like lactic acid, acetic acid, and formic acid as an end product. And based on the end product formation, we can confirm what organism, at what enteric organism it is. And in methyl red test, the bacteria has grown in a broth called MRVP broth. And if the bacteria has the ability to utilize the glucose with the production of stable acid, the color of the methyl red, it changes from yellow to red color when added into the broth culture. And thus the requirements is MRVP broth called methyl red August Proskauer broth, and methyl red indicator, inoculating loop which is made up of nichrome, etc. So when you inoculate, what is happening during the 24 hours? So it can be incubated for even 24, 48 or 72 hours. And after that, when you add 5 drops of methylate indicator to the broth, so the glucose, if it is utilized by the organism, based on the nature of organism, it will be producing some stable acids like lactic acid, acetic acid, and formic acid. So it will be remain in the medium for longer duration, and it will be in the pH of 4, so that it produces a red color. It indicates that the positive reaction for methyl red test. If this acid, what it is produced is unstable acids, or it is not utilized by the organism, so it produces yellow in color. So that is called as methyl red negative test. So in general, positive, it is a indole methylate positive is a red color, and methylate negative, it is yellow in color. So this is a rough indication about the stable acid. So acidic pH, the stable, when it goes for less than 6, it will be of acid, and it will be of the pH 4.2, and that it produces the red color. When the pH is more and there is no change in the color, it will be remain as yellow color. That is called as the methyl red negative. And E. coli, it produces stable acid by utilizing the glucose, and both Klebsiella and Enterobacter aerogenes, it won't produces any stable acid after utilization of glucose. So this is second confirmation, both in indole and methylate. When you are getting a positive reaction, you can say the organism, it may be belongs to the genera Escherichia coli. Now coming to the next test in the IMViC test called Proskauer test, and this is one that is to determine the ability of the organism to produce the neutral end product called acetyl methyl carbonyl. In short, it is called acetine, and it is also utilized or it is produced by glucose fermentation. So in this, the organisms I have taken Klebsiella or Enterobacter for our understanding, and these two it produces a stable acid, the product of acetine, and this will be the chief end product of the glucose metabolism, and formed a mixed acid. And later, this acetine can be oxidized into diacetyl by adding alcoholic alpha naphtha. It serves as a catalyst to bring out the red complex. So this is what the principle behind in the Voges-Proskauer test, and both methyled and Voges-Proskauer can be tested a medium called M or VP. A same media can be utilized it for both the test. And in this reagents what we are adding after 24 or 48 hours, 5% of alcoholic alpha naphtha plus 40% of potassium hydroxide. So during this addition, so after 24 hours, when the 37 incubation at 37 degrees Celsius, 1 ml of a broth into sterile test tube and add zero point 0.6 ml of alcoholic alpha naphtha and 0.2 ml of potassium hydroxide, and shake the test tube and expose the medium to an atmospheric the oxygen and allow the tube to remain undisturbed for 10 to 15 minutes. And based on the color change, and we can confirm what organism it is. So this is what the chemical reaction it is happening during the 24 hours or 48 hours of incubation. And the produced acetylene, when you add alcoholic alpha naphtha, it produces a diacetyl and guanidine group complex, and that it produces the pink color complex. This is the reason for the Voges-Proskauer color change after 24-hour, 48-hour color change. So here the positive test, it indicates the presence of diacetyl, and the oxidation product is called acetine. It produces the pinkish red color at the surface of the medium. So here also the color, it won't be produced in the whole medium. So it will be after exposure of the oxidant 10 to 15 minutes of the alcoholic alpha naphtha and potassium hydroxide in the test tube B, it produces the positive color, and Voges-Proskauer test is negative for E. coli, even though it has produced a positive test in both indole and methylate. Once again, you have to perform it in Voges-Proskauer test. By the time when you add this alcoholic alpha, naphtha and potassium hydroxide, when there is no color change, it is a rough indication. Can indirectly you can confirm it as E. coli, and meantime we can confirm it's Enterobacter aerogenes. And VP positive, one best example is Enterobacter and Klebsiella pneumoniae. And VP negative, it is Escherichia coli. And finally, citrate utilization test is another interesting test. And this test is based on the ability of organism to use citrate as the sole source of carbon. In the medium, when there is a carbon source is absent, only few genera of organism is able to utilize citrate as the sole carbon source by secreting an enzyme called citrate permease. But every organism, all the genera doesn't have this ability. So based on the utilization of citrate, we are confirming what genera or what enteric bacteria it is. So in this, the principle behind the citrate utilization test, the test organism is cultured in a medium which contains sodium citrate only. In the previous three tests, it is containing the medium of glucose or other carbon source. But in this particular test, we made the medium which is lack of glucose. It will be only a source of citrate as a sole carbon source. Additionally, it also contains some ammonium salt indicator and bromothiamyl blue. And bacteria that is able to grow in this particular medium by seeing the environment, by seeing the substrate of citrate, it is able to secrete an enzyme uh, called the adaptive enzyme. So there are two types of enzymes always within the organism. One that is adaptive and constitutive. Constitutive enzymes always it will be present within the bacteria. It required for regular metabolic activity. Adaptive enzyme depend upon the environment, what type of substrates are available. Accordingly, the organism it starts secreting some enzyme. So this adaptive enzyme production, only few genera of Enterobacteriaceae is possible. So one such an enzyme called citrate permease will be produced only by few genera. and those organisms is able to produce a citrate permease, is able to allow the citrate into cell wall, and so that it converts citrate into pyruvate. So this color change or this organic compound conversion we are confirming by the pH color change. This test we are performing in citrate agar medium. In the previous three tests, so after 24 hours or 48 hours of incubation, we are adding some additional reagent, but this test directly we are performing in citrate agar medium, and that is the reason it is called as indole methylate August Proscar in citrate agar medium. That's the reason the IMViC already in the first slide I was explaining. And now the growth is shown by a change in the color from light green to Prussian blue color, and due to the alkaline reaction. So what is that chemical reaction? I'll explain you in the next slide. And if the citrate is utilized, so when the media is utilized, the media what we are used here, it is the Siemens citrate agar medium. And here the general requirement is a 24-hour to 48-hour old culture of Klebsiella pneumoniae and Siemens citrate agar medium, which will be serves lack of glucose and only citrate as a sole source of carbon, and Bunsen pharma for maintaining the aseptic conditions and inoculating needle with nicro. And this is what the actual reaction. The right hand side is a Prussian blue color, the other one it's in light green in color. So that is what the actual color. The right hand side, the green in color, that's what the actual color of the medium. So when the inoculum is streaked and then when you maintain to 24 to 48 hours, so the color change from green to Prussian blue, that indicates that the organism is able to secrete an adaptive enzyme called citrate permease. So the actual reaction, this is what is happening. The Siemens citrate agar medium, which will be containing the citrate along with water. If the organism is able to produce the adaptive enzyme of citrate permease called citrase, it will enter into the cell wall and directly it enters into the TCA cycle, and it produces the oxaloacetic acid and the acetic acid. Later, this oxaloacetic acid it gets converted into pyruvic acid, and also it produces some carbon dioxide. And this produced carbon dioxide that combines with sodium. Sodium it is already as an additional inorganic compound in the Siemens citrate agar medium. This produced carbon dioxide and sodium combines together and it forms a compound called sodium carbonate. This sodium carbonate is the reason for the medium from acidic medium into alkaline medium. The bromothimal blue, it produces the Prussian blue color in the alkaline medium. So finally, this alkaline pH can be detected by using the color change. So that indirectly we are confirming whether the organism is able to produce citrate permease or not. So only few genera, especially Klebsiella pneumoniae, is having this ability to produce a citrate permease, and so that we are able to identify or differentiate the enteric organism by using this IMViC test. So this is what the reaction actually it will happen. A growth with the color change from green to Prussian blue color along this land, and that is called the organism is utilized or is citrate as a sole carbon source by secreting an enzyme called citrate permease. And when there is no growth as such, the medium it remains the same green in color. So that indicates that the organism doesn't have the ability to secrete an adaptive enzyme called citrate permease, so that we can confirm definitely the organism is what genera it is. And the given culture has the ability to utilize the citrate as a sole carbon source if the color change is changed from green to Prussian blue color. And here the examples the citrate positive on genera is Klebsiella species, and citrate negative is Escherichia coli. So Escherichia coli, both indole and methylate will be positive, and both August Proscar test and citrate utilization test will be acting as a negative. So this test I just explained in a simplified manner to understand quickly what is this IMViC test. And this is a repeated question in most of the universities, and as well as both theory as well as in practical. And by the time of explanation, if you explain with the chemical reaction and a colored diagram, including this indole production with the cherry red color on the top layer, and after the color, after the chemical reaction with the colored diagram, and with the explanation of these three organisms, that is very much sufficient to get full or good marks. So thank you for listening.